Forensic cancer research is really a unique group in terms of bringing experts from academia, from the regulatory agencies, from pharmaceutical, and including patient advocates, bringing us all together to talk about these important issues. And I was amazed not just what I learned from the scientists in the group, but also the give and take and the amount of thought that needs to go into this.
What we wanted to do with Friends of Cancer Research is beyond just sort of day-to-day use of ctDNA in the clinic. It's asking the simple question, can ctDNA measurements in clinical trials tell us about whether a drug is effective much earlier and much more accurately than we currently get that type of information? Traditionally, for example, you might test a drug in a population of individuals and look at, say, overall survival or time to recurrence on imaging. But potentially with ctDNA measurements, you could get this information really early, say within 3 weeks or 6 weeks of starting treatment. But that at the moment is just a potential.
Patients are desperate for new, effective, and safe drugs to treat advanced cancer, to treat all cancers. And anything that we can do to bring those treatments to market sooner is something that we really need.
So what we wanted to define with this white paper and this working group effort is, what do we need to do as a community to prove that ctDNA is an early endpoint that can give us all that information? So that's actually quite a complicated thing to do. You need data across lots of different clinical trials, and that data needs to be standardized in such a way that you can compare across it. So what we really discussed in this working group was, what variables do we need to standardize? And it can be quite simple things like, we need to make sure that across trials we collect blood at the same time points so that we can compare across time points. We need to make sure that we use the same type of assays to measure how much ctDNA there is. And all this together will allow us in future to actually do a large meta-analysis and then look across all the different trials, across different cancer types and treatments, and say, yes or no, ctDNA is validated as an early endpoint in clinical trials.
What struck me, and I think what advocates would relate to, is that there is a difference between the use of ctDNA in the clinic when we may be looking at someone's progression of diseases or recurrence or what's happening there, as opposed to in a trial like this, a prospective trial, where we're really looking at response to treatment. So there's a little difference there, and people who are in the trial may never really know what their results were. They may, but they may not, because this is for regulatory purposes. The other thing was how there is a balance between needing to have a uniform framework for these prospective trials in terms of when data is collected and how it's used and the assays, how they're related, et cetera. But also there needs to be some flexibility, because with baseline and when people receive treatment, there can be some differences. It can be 3 weeks, 4 weeks. There may need to be a holiday for treatment for various purposes. And so to make it flexible enough to fit patient treatment schedules, but also uniform enough to fit the clinical trial, is a real balancing act.